Good morning, everyone, and welcome to today's webinar hosted by Gator Bio in collaboration with NEBA Labs, titled Novel Strand-Specific Approach for AAV Genome Titer and Genome Integrity Determination using DNA Hybridization and Biolayer Interferometry. My name is Chris Lalu. I'm a Marketing Communications Specialist at Gator Bio, and I will be moderating the webinar today. Before we get started, I'd like to go over a few housekeeping items so you know how to participate in today's event. All attendees are listening using your computer speaker system by default. All the attendees will be on mute by default, and the in-meeting chat option is disabled. However, you can message the moderator at any time in case you're having any technical difficulties. One will have the opportunity to submit text questions to today's presenters by typing your questions into the questions pane of the control panel. You may send in your questions at any time during the presentation. We will collect these and address them during the Q&A session at the end of today's presentation. Getting into the format of this webinar, we will have both speakers present for about the next 25 to 30 minutes. And once they finish, there will be a quick poll on your screens that will ask you to participate in, followed up by the Q&A session. So let's begin. I would now like to introduce our speakers for today, Vasundhara Bahel, Product Manager of Consumables at Gator Bio, and David Dobnik, Co-founder and CSO at NEBA Labs. Handing it over to Vasundhara, our first speaker of the day.
Hello, my name is Vasundhara Bahel, and I'm a product manager at Gator Bio. Many of you are familiar with Gator, but for those who are not, the following slides will give you an overview about the company. So this will be a very short introduction before I hand over to our guest speaker from NEBA Labs. Gator Bio specializes in the label-free Biolayer interferometry technology, and we are based in Palo Alto, California. We offer a complete BLI package that includes instruments, consumables, and software. We have a very strong IP portfolio with over 50 granted patents, and our customer base is rapidly expanding all over the US and worldwide. Our R&D teams and the teams for biosensor manufacturing are based in Palo Alto, and we have a team of dedicated field application scientists, service engineers, and sales professionals offering support to our customers all over North America. Our BLI applications include quantitation, kinetics, and epitope binning for the antibody therapeutic development. And we also have products for viral vector analytics and lipid nanoparticle analysis in the field of gene therapy. All our products are based on the principle of biolayer interferometry. So exactly what is biolayer interferometry? It is a label-free technique for studying biomolecular interactions. BLI detects interactions by measuring the interference pattern of white light reflected from a biosensor surface coated with ligands. When target molecules bind to these ligands, they alter the optical thickness of the layer, changing the interference pattern. Real-time monitoring of these shifts allow precise analysis of association and dissociation rates among the ligand and the analyte. This can be used to quantify analytes or conduct an affinity analysis between biomolecules. So Gator Bio offers a complete solutions package, including instruments suited for different throughput, biosensors for various applications, and a software for data acquisition and analysis. We have 5 different instruments at this point to suit different throughput needs, starting from 4 simultaneous readings to 32 simultaneous readings. The instruments are all fluidics free, making them low maintenance, and they are compatible with crude samples such as supernatant, cell lysates, and plasma. So they are very well suited for in-process samples, which are usually in complicated matrices, as well as to purified samples in more simplified buffers. This slide shows the 5 Gator instruments, with Pilot being the lowest throughput, offering only 4 simultaneous reads, all the way to Gator Pro, which is the highest throughput instrument that lets you analyze 32 samples in parallel. And it can analyze 1152 samples per batch. Coming to our biosensors and kits, we have roughly 28 at this time. These include sensors for unlabeled antibodies, for biotinylated proteins, small molecules and peptides, tagged proteins, and viral vectors. Some of our recently launched novel products include the streptactin biosensor for twin strep tagged proteins and anti-VHH biosensor for camelid nanobodies. As for gene therapy, our suite of products includes products for capsid titer and a product for AAV empty and full ratio analysis. Then coming to the Gator 1 software, this is the tool for data acquisition and analysis. It's a very straightforward software to use and offers real-time monitoring of the changes in interference patterns. It's easy to use and is 21 CFR Part 11 compliant. Gator also offers low-binding BLI plates in 96 and 384 well formats. Tilt versions are available now that allow for reduced sample volume, helping conserve precious samples. So in summary, Gator enables various applications like quantitation, kinetics, and epitope binning with a variety of analytes ranging from antibodies to tagged proteins and peptides, as well as viral vector analysis like AAV titer and percent-full ratios. We support different throughput needs through a range of instruments that are low maintenance and easy to use. We also support our users with custom biosensors for their niche projects, for which more information can be found on our website. With this, I'd like to conclude the introduction to Gator and hand over to our speaker from NIBA Labs. So over to you, David.
Thank you, Vasu, for this nice introduction. So, as said, I'm David Dobnik, and today I will present you the topic of genome titer and genome integrity quantification using the novel approach by Biolayer interferometry. Just legal disclaimer that the views that I will present here are exclusively mine and are not the Gator Bio views, and the work presented was also financially supported by Gator Bio. The presentation will include introduction on viral vector characterization so that we see why the characterization of the capsid content is important. Then I will move to the quantitative perspective on the capsid content characterization focused on genome integrity, mentioning options, but the main point of the case study that I will present will be the use of Biolayer interferometry for genome titer and also for genome integrity. In the end, I would have some few conclusion points, and I hope that we will have a nice Q&A session as well. But first, just a short introduction of myself. I've been working in the field of gene therapy since 2016, mostly on the developing of methods for characterization of viral vectors. I'm a co-founder of NIBA Labs, Slovenian life science spin-out from NIB, with mission to improve speed and efficiency of process development in the cell and gene therapies by really providing an expert service to our partners. Now, let's move to the gene therapy as such. So, the gene therapy is all about delivering some construct that will help to cure a disease or make a change in the genome to the targeted cells. But it's not just important that we do deliver something into the cells, it's also what we deliver. And viral vectors, as delivering the payload to the cells, they need to be well characterized so that we really know what they deliver and that we can be sure that the patient will have benefits from the delivered payload. For this purpose, there's a plethora of analytical methods that can and must be also used for product characterization. and those methods are addressing different critical quality attributes. And today we'll be focusing mostly on the vector genome, for which currently, let's say, there are qPCR and digital PCR methods that are mostly used for this purpose. There can be different things that are present within viral vector capsids, not only the intended full-length genome, but also we can have fragments of the genomes, we can have empty particles, there can be some plasmid fragments included, or even like whole cell DNA fragments. But anyway, within the population, we have different things that are in the capsids. It's almost never that we would only have, let's say, the intact genomes that we want. And actually, just until recently, let's say a year or two ago, the focus on the capsid content was to identify full and empty capsids as such. Then recently, we came into the idea that there are also some partials, so-called partial particles that include some fragments and appear somewhere between the full and empty ones. And of course, to analyze that, there's again a variety of methods that can analyze the full, empty, and even sometimes partial capsid content. But each of these methods has its own challenges as well. And beside that, they're also not performing equally well. And which method to use depends on the process, the established process that you have in-house. But you must be aware that you might have some bias using a specific method. Another problem that I see with those methods is the resolution. So what is actually included in the width of this peak? And to show you that, I would just like to use the example of this chart where the claimed resolution of the method is 150 base pairs. But if we just check the distance between the middle peak and the larger peak, you can see it's like 2.4 kilobases. And if we translate this distance, although the relationship is probably not really linear, to the width of this peak in the middle, it's somewhere around 1.2 kilobases. Now, if we go further and move that into the AAV genome, you can see that there are some, let's say, initial construct parts that are relatively short, but when you come already to the promoters or to the terminators, the polyA tails, you can have quite a variety here. And if the peak includes, let's say, 1.2 kilobases of sequences, you can see that you might be including in this peak also some fragments of the construct as well. So just to put it schematically a bit in a bit different way, any of these peaks can include different things. So even the full particle peak can probably include some fragments. And also, since these methods are unspecific, you can quantify full particles also if they are full with some other things than intended construct. So to actually have a better insight into what is in the capsid so that we don't find some surprises hiding there, we need to use the methods that can provide actually vector genome specific information. And integrity is actually becoming a really important quality attribute in this fashion, since not only that we just quantify the amount of genomes, we need to show that it's there present in its full length. And there is an increasing number of analytical approaches already to characterize the capsid content. Also, publications addressing genome integrity are piling up. There is also increasing number of companies researching genome integrity of their pipeline products. But there are limited possibilities for quantitative target-specific methods to evaluate genome integrity. Probably all of those who are working in this field are familiar with long-read sequencing, which can give relatively good information on the integrity of the sequence. However, the quantitative aspects is still, let's say, slightly biased due to some issues of, let's say, sequencing bias against shorter reads. Then there's digital PCR, multiplex digital PCR, that is relatively widely used as well for this purpose of genome integrity. And it gives a really absolute quantification of the fragments that are present in the capsids. However, there is one aspect that digital PCR is not covering, and this is the specificity for individual strands, because we have single strands in the genome of the AAV genome, and you don't know whether you have plus strands, minus strands in the same ratios or not, and if there might be some bias there as well, which could help, let's say, with some optimization. And here we do have a new approach, that Biolayer interferometry, that actually can help also with this strand-specific information. So until now, as presented also by Vasu, the Biolayer interferometry was used for evaluation of different quantities of proteins, some evaluation of protein interactions or molecule interactions. And the principle, as described, is the way that whenever you have binding of the analyte to the probe, there is a shift in wavelength, and you can actually then use this shift to have a standard curve and also make some quantification of the intended binding. And with the Gator's BLI system, now we also went into the field where we can quantify nucleic acids without any amplification. And this is actually performed in a strand-specific way in a simple three-step procedure, including lysis and hybridization, detection, and analysis. All of the results that I will show in this presentation were generated with Gator Plus system from Gator Bio. So to go into more details of this three-step procedure, so the first step, which includes lysis and hybridization, is actually pretty simple. So you have a sample that is usually, let's say, DNA-treated sample to remove all the free-floating DNA. You add lysis buffer, oligos that will bind to the specific target region, and also some additional biotinylated polyA oligo. You heat the sample to hybridize oligos to the DNA that will be exposed, cool it down to 4 degrees, and in such way you get this hybridized complex of oligos and the target of interest. What follows detection and analysis steps, where we take the hybridized sample pipetted to the well plate together with the reagents and the substrate. We put it in the machine and we get the binding to the probes from the fluorescein part with using anti-fluorescein biosensor, and the other oligo acts as a target for biotin detection that helps improve the signal intensity. After that, the probe with bound complex is measured for the wavelength shift, and this wavelength shift is transformed to the standard curve, which can be used for quantification of the sample. First, I can tell you that we started working on the vector genome titer with this approach, and we have seen very promising results using the BLI assays in terms of repeatability and quantification, which we showed on different AAV serotypes. So we tested AAV 5, 8, and 9, and we used positive and negative strand-specific assays. You can see that the shapes of the curves are similar. However, the wavelength shift is not the same for all assays. But this is not problematic because these points are used to generate a standard curve, and since the sample then behaves in the same way, these differences between the positive and negative strands or even serotypes are really not important. So here I have the results. So first results that I would like to show that are on the standard curve samples, where we've tested it over different days. And you can see that the variability was pretty good in the expected range. So definitely this was a good starting point to show that we can use this kind of standard curve for sample quantification. And then we tested 3 unknown samples, although we had the expected concentration that was calculated. And you can see that we've come relatively close to the expected concentration, at least with these 2 samples. At higher concentration, the difference was slightly higher, but this is just because the standard curve variability here at this level was also slightly higher. So since this approach is actually quite good just for the simple titer quantification, so strand-specific titer quantification, we said, why not take this further to quantify vector genome integrity? So if we just have the short area where we have the 2 oligos targeting this part, we could perhaps do that on the longer target area. And by longer, why not do the whole length AAV genome? And actually, this approach was just transferred to the steps that we already have. So the lysis hybridization, detection, everything was the same, just the position of the oligos. So the oligos were different. And here I have the results of the standard curve for negative and positive strand-specific genome integrity assay now. So this is not just the specific target like it was before, like CMV. Now it's the whole genome. And you can see that the shape of the curve is actually pretty much the same as it was with the genome titer. assay, what you might notice is slightly higher variability in the response that we got with at the highest concentration, and this is for both negative and positive strands. And based on those standard curves that I've shown, we have also made some quantifications of unknown samples, again with some expected concentration that was calculated based on the multiplex digital PCR. And here are actually the preliminary and probably the first vector genome integrity results that were published or at least made public in terms of BLI system. As you can see, the results are very encouraging, especially this intermediate concentration is actually almost the same as expected. Very low small difference in terms of what we expected and what we got. Highest concentration based on the variability of the standard curves. Here we got a slightly higher difference to the expected concentration for both negative and positive strand. And the CV that you might notice here seems to be high. However, if we check the individual runs, there is, let's say, sometimes not so much variability, sometimes there is slightly higher. But I must tell you here that these are really not truly fully optimized conditions. So this is just the proof of concept that we can also evaluate vector genome integrity with the BLI system. So why the variability? I did not mention that before. Since we have a longer DNA strand, there is a chance, of course, that this strand takes some form of, let's say, a shape that is like a small ball, so that the signal is closer to the probe, or it can be a bit more relaxed or stretched, so the signal is a bit farther from the probe. And that is the reason why we might get a bit higher variability at the level of the genome integrity assay. But it's not just for full-length evaluation. If we position oligos on different regions of the genome, we could essentially make quantification of the fragments that might be present in those capsids as well. So definitely there is some very good, let's say, starting points on which we could build with this assay. And the advantages are definitely the strand-specific quantification of, let's say, any kind of defined vector genome length without amplification. So it's really almost like going directly into the sample, not amplifying DNA. We have quite repeatable results in relatively short time. So the shortest time to result is 30 minutes. But of course, there are still some things that need to be addressed with further experiments, like establishing the most suitable standard curve. What I've shown you before was the standard curve that was prepared based on the AAV sample, but we would like to take this assay further by preparing really a well-characterized material that could be used for different purposes and would give good information on all of the strands, so positive and negative strands. Then what we need to do is also test the different combinations of the assays and then show, let's say, this fragment content comparability to the results that we have already with the multiplex digital PCR. Of course, the comparison is slightly challenging because of the strand specificity. However, by using the well-characterized standard material, we might get even more information from the BLI assay. And in the end, of course, we would need to thoroughly evaluate the performance and perform the qualification of such assay. So that brought us to the take-home messages, and I hope that I've shown you that it's really that we need to move just from the full empty evaluation to go to the specific capsid content. So this was from the introductory part, and for now there are only few methods that enable quantitative evaluation of vector genome integrity, and BLI is of course one of those few. What is important with BLI is to use a well-characterized reference samples because it's still a method that relies on the standard curve. BLI results can be obtained in a very short time, at least compared to other methods. And this novel assay can actually enable strand-specific quantification, which is not possible with PCR. Also for the vector genome integrity, and this is done without DNA amplification. So with that, I would like to acknowledge my colleagues who did a lot of work in the lab on that, and also Gator Bio for all their support. And with that, I would like to open the webinar for the Q&A session. Thank you.
Thank you, David, for a great presentation. Before we get into the Q&As, I'm going to launch a really short poll on your screen. Please select the appropriate response and click Submit. So the first question in the poll says, what do you use for AAV Genome Titer? It's multiple choice. Go ahead and select whichever one applies to you. I'll just take a moment there. The next question asks, how many samples do you analyze in a day? Listing various throughputs. What is your pain point? A few different options there. So just take a moment to go ahead and select one of those. What methods do you use for genome integrity? And then lastly, an option if you want to be contacted by someone on sales or from Neva Labs, Gator Bio, etc. So I'll just give you guys about 20 seconds or so, quickly answer those, and then we'll end the poll and proceed with the Q&A. Okay, just a few more seconds. It looks like most of these are populating. Okay, it looks like most of you have responded. Those of you who have not yet responded, go ahead and submit your responses now, and we'll close the poll in just a moment. Okay. Thank you, Vasundhara and David. We're now going to begin answering the questions submitted during today's presentation. As a reminder, you can still submit questions through the questions pane in your attendee control panel. And it looks like we already have a few questions here. Give me just a moment. Okay, so the first question is, does the standard curve need to be plasmid DNA, or can AAV gDNA be used?
Yes, I perhaps slightly answered this question after it was said,
David, I think you're
after it was typed. But anyway, as mentioned, we've used only AAVs in our example as a standard, so it can be AAV DNA. However, now, as said in the end, we would prefer to go into the more controlled format, perhaps like plasmid DNA or even kind of synthetic DNA, especially from the strand-specific way of quantifying, because with AAVs, we are actually not certain what's the amount of each of the strands that are there. So what we took for our examples was just kind of an average and the value from digital PCR, considering, let's say, we have 1-to-1 ratio. But if you have a plasmid DNA as a standard, you always have 1 plus, 1 minus. And if you have it well characterized, you can then really get this kind of strand-specific information. And when we were doing these different AAV samples, we also tried using plasmid DNA. And there we have seen that for some serotypes, the ratio of positive and negative strand that was packed was different. In some it was half-half, but in some it was more like 70-30. So it's important what you use as a standard curve as such.
Okay, great. Thank you. And another question here. Is the biotin polyA substrate necessary to see a large enough wavelength shift? Would the assay work without it?
I can answer that. So yes, it is necessary. So the way it is optimized, it does rely on immunochemistry-based signal amplification. So the assay is not going to work without the substrate.
Okay. And follow up, do you have any thoughts on doing RNA hybridization with BLI?
Yes, we do. So that's in very early stages, and it's not on way to being a product yet. But yes, we are looking into it, and we do have plans of doing that.
Okay, great. Let's see a few more questions rolling in here. How much sample is needed, and what is cost difference with BLI versus multiplex dPCR? What is the sensitivity compared to multiplex dPCR?
Sample requirement is about 10 to 20 microliters. And the cost difference, the cost will be comparable to PCR, most likely lower. And the sensitivity, it goes down to 1E9 Vg per ml. And the range is 1E9 to 1E12 Vg per ml.
Thank you. And a few more questions here. This one may be for NEBA Labs. Can you comment on the applicability of BLI for genome integrity testing for self-complementary AAV genomes?
Yeah, that's quite an important question. Since integrity of self-complementary constructs cannot be assessed by multiplex digital PCR just because of the complementary targets on each of the sites and the principle of PCR. However, with the BLI system, this should, let's say, at least in theory, work. I must admit that we did not try it yet, but since it is a strand-specific technique, you have different oligo targets in both of the strands, although they are complementary. So in theory, I would say that it could be used also for that, and perhaps one of the only technology that can give you some kind of quantitative evaluation of that self-complementary vectors.
Thank you, David. And another question here. What is time to results using Gator's product for capsid titer?
The product for capsid titer relies on direct binding, and you can analyze 8 samples in parallel in 2 minutes. And for very high titers, it's as quick as 30 seconds. And depending on the instrument, it can either be 8 or 16 or 32 samples in parallel. So that will be, if it's a very high titer ranging 1E12-ish, then it's about 30 seconds. If it's lower titers, then 2 minutes.
Okay, thank you. And looks like another Gator question. Are Gator's AAV products compatible with crude samples?
Yes, they are. So most of our, actually all of our products for capsid titer as well as the ratio, and then our upcoming product with genome titer, they're all compatible with crude samples. And I just want to point out there's resources on our website. We have a white paper published with one of our collaborators that explains how our assay performs in crude samples for empty and full ratios compared to other orthogonal techniques.
Thank you. And this looks like a question for David. What would be the best choice for standard curve material?
Well, as I've already said before, in my view, it would rather, of course, be some controlled material. But then on the other hand, if you have a material for the standard curve that is not the same as your testing material, you might have some bias. So a bit more of the evaluation of such material would be needed. But as said, if you're using AAVs, you might not be completely sure how much of those, let's say, specific strands you have inside. So my preference would be a bit more controlled material like, let's say, fragments of DNA, double-stranded DNA.
Thank you. And just checking if there's any other questions here. We'll give it a moment. It doesn't look like there's any more. So with that said, thank you to our speakers, Vasundhara Behel and David Dobnik, and thank you everyone for attending today's webinar. If you have any other questions, please contact us at info@gatorbio.com. Once you leave today's webinar, you will receive a follow-up email within 24 to 48 hours with a link to view a recording of today's webinar. So on behalf of Gator Bio, NEBA Labs, and our presenters, thank you for joining us today and have a great rest of your day.